Polypterus is a genus of freshwater fish in the bichir family, Polypteridae, of order Polypteriformes. The type species is the Nile bichir. Bichir fishes in this genus live in various areas in Africa. The etymology of the genus name derives from a combination of the Greek prefix pi omicron lambda epsilon, poly, many, and the root word pi tau epsilon rho omicron nu, pteron, wing a fin. Many fins. Polypterus is the only known vertebrate to have lungs, but no trachea. Recoil aspiration. In shallow water, Polypterus inhales primarily through its spiracle blowhole. Exhalation is powered by muscles in the torso. During exhalation, the bony scales in the upper chest become indented. When the muscles are relaxed, the bony scales spring back into position, generating negative pressure within the torso, resulting in a rapid intake of air through the spiracle. The air is nearly sufficient to fill the lungs. This is followed by one cycle of buccal, mouth, pumping, which tops off the lungs, with the surplus air from the buccal pumping process discharged through the pharynx. According to one hypothesis, Devonian tetrapods may have inhaled in this way. Discovery. Polypterus was described, and named in 1802 by Etienne Geoffroy Saint-Hilaire. It is a genus of 10 green to yellow-brown species. Naturalists were unsure whether to regard it as a fish or an amphibian. If it were a fish, what type was it? Bony, cartilaginous, or lungfish? Some regarded Polypterus as a living fossil, part of the missing link between fishes and amphibians, helping to show how fish fins had evolved to become paired limbs. In 1861, Thomas Huxley created the order Crossopterygi to house animals, fossil and living, that possessed lungs and fleshy pectoral fins with lobes. He placed Polypterus and Calamoides within this order, allocating them to a new tribe, Polypteridae, which he created especially for them. The weight of Huxley's authority allowed this allocation to last in textbooks and lectures long after it had been disproved. In the 1870s and 1880s, Francis Balfour and his students had shown that embryology could help to answer questions about the evolution of species. No one had studied the embryology of Polypterus. Someone who could do this might prove the missing link theory and be greatly honored. But it could be a dangerous quest. The only breeding Polypterus specimens were in swampy parts of African rivers. Africa was a turbulent place and swamps were rich breeding grounds for mosquitoes carrying malaria. Two men, Nathan Harrington and John Samuel Budgett, attempted to answer this question by making repeated expeditions to Africa. Harrington failed on his first attempt in 1898 and died early on his second in 1899 before he could reach his destination. Budgett failed in 1898 1900, and 1902. He finally succeeded in 1903, but died of blackwater fever shortly after his return to England. He left excellent samples and drawings, but his only writing was a diary. Consequently, his results on Polypterus were written up and published by his friend John Graham Kerr. Drawing on this work. In 1907, E.S. Goodrich reported to the British Association the then current state of evidence against Polypterus being a Crossopterygian, placing it within the Palaeonichids, the most primitive Actinopterygians. Much later, in 1946, Romer confirmed this view, but he also wrote, "The weight of Huxley's 1861 opinion is a heavy one," and even today many a text continues to cite Polypterus as a Crossopterygian, and it is so described in many a classroom. Although students of fish evolution have realized the falsity of this position for many years, Polypterus is not a Crossopterygian, but an Actinopterygian. and hence can tell us nothing about crossopterygian anatomy and embryology. Quote, Hall, 2001. Relying on Patterson, 1982, and Nowak al. 1996. Writes, phylogenetic analyses using both morphological and molecular data affirm Polypterus as a living stem actinopterygian. Research is ongoing. Most of the conclusions drawn by Kerr from Budget's specimens have been confirmed, but many questions remain. Polypterus have rarely been bred in captivity. The first success was that of Polypterus senegalus by Arnault in 1964. A species spawned repeatedly since. See Hartle, 1981. Barch and Gembella, 1992. Barch al. 1997 and Shugart, 1997. Shortly after Arnault's success, a second species, Polypterus ornata was spawned by Armbrust for the first time, 1966 and 1973, and bred subsequently by Azuma in 1986. Wolf, 1992. Barch and Britz, 1996. The third species successfully spawned in captivity was Polypterus endlicheri by Azuma in 1995. Zubazal have been successful in breeding Polypterus in captivity. In December 2005, several eggs were laid, and at the beginning of 2006, 6 young had hatched. Within 2 months, they reached 10 centimeters, about 4 in. In 2014, researchers at McGill University, published in the journal Nature, turned to Polypterus to help show what might have happened when fish first attempted to walk out of the water. The team of researchers raised juvenile Polypterus on land for nearly a year, with an aim to revealing how these terrestrialized fish looked and moved differently. Video. Species: Polypterus ansorgi Boulanger, 1910. Guinean bichir. Polypterus bichir lacipede, 1803. P. B. Bichir lacipede, 1803. Nile bichir. P. B. Katangai pole, 1941. P. B. Lapradii Steindachner, 1869. Polypterus delhezi Boulanger, 1899. Bud bichir. Polypterus endlicheri Heckel, 1847. P. E. Conjicus Boulanger, 1898. Congo bichir. P. E. Endlicheri Heckel, 1847. Saddled bichir. Polypterus mokalembe Mubschliwen and Schaefer, 2006. Mokale mombe bichir. Polypterus ornata Boulanger, 1902. Ornate bichir. Polypterus parmasses, 1850. P. P. Bueticoferi Steindachner, 1891. Bueticofers bichir. P. P. Parmasses, 1850. Shortfin bichir. P. P. Poly J. P. Goss, 1988. Poles bichir. Polypterus retropinus valent, 1899. West African bichir. Polypterus senegalus Cuvier, 1829. Grey bichir. P. S. meridionalis Pol, 1941. P. S. senegalus Cuvier, 1829. Polypterus Tugelsii Britz, 2004. Cross River bichir. Polypterus weeksii Boulanger, 1898. Mottled bichir. Sources: Polypterus. Integrated Taxonomic Information System. Retrieved 4 May 2005. Froese, Rayner, and Daniel Pauly. eds. 2005. Species of Polypterus in Fish Base. May 2005 version. Goodrich, E. S. 1908. On the Systematic Position of Polypterus. Report of the 77th Meeting of the British Association for the Advancement of Science. 1907. 545 to 546. Quoted by Hall. Hall, B. K. 2001. John Samuel Budgett, 1872–1904. In Pursuit of Polypterus. Bioscience. Volume 51. No. 5. May 2001. pp. 399–407. Kerr, J. G. 1907. The Development of Polypterus senegalis CUV. Pages 195–290 in Kerr, J. G. ed. 1907. The work of John Samuel Budgett, Balfour student of the University of Cambridge, being a collection of his zoological papers, together with a biographical sketch by A. E. Shipley, F. R. S., and contributions by Richard Ashton, Edward J. Bless, Edward T. Brown, J. Herbert Budgett and J. Graham Kerr, Cambridge, UK, Cambridge University Press. Noack K. Zadoya R. Mayer A. 1996. The Complete Mitochondrial DNA Sequence of the Bichir, Polypterus annati pinnis, a basal ray-finned fish. Ancient Establishment of the Consensus Vertebrate Gene Order. Genetics 144. 1165–1180. Cited by Hall, Patterson C. 1982. Morphology and Interrelationships of Primitive Actinopterygian Fishes. American Zoologist 22. 241–260. Cited by Hall, Roma, A.S. 1946. The Early Evolution of Fishes. Quarterly Review of Biology 21. 33–69. Cited by Hall.